My research interests are concerned with the study of the structure and function of membrane proteins. Membrane proteins are amongst the most important proteins in the cell because they facilitate the communication between inside and outside of the cell, but they also facilitate the communication between the cell and the environment. Furthermore, they are also important because a quarter of all genomes encode for membrane proteins, and 70% of the drug targets target membrane proteins. Part of my research addresses the fundamental questions of life and investigates, for example, how electrons are transferred within proteins. The questions are directly linked to how certain diseases occur. And the atomic models of membrane proteins are the basis for structure-based drug design. And this way we can develop drugs with higher specificity and less side effects. My research is funded by the Science Foundation Ireland, by the Irish Research Council, and also by the European Union framework. So, yes, over the past 2 decades, my active research has made a major contribution in the field of bioenergetics. We address the questions as to how thermophilic organisms deal with molecular oxygen. For this, we use a combination of biochemical, biophysical, and structural approaches to study the function of membrane proteins. The recent achievement, which was solving the crystal structure of the cytochrome C oxidase, which is called CA3 oxidase, was a highlight of my research. And this earned a spot in the best scientific journal, which is Nature. So working on membrane proteins and structural biology of membrane proteins is very hot business in the scientific world. For example, for the structure of the beta-adrenergic receptor, G protein coupled receptor, has led to the Nobel Prize in chemistry last year. So it is quite a hot area, and any publication that comes in terms of novel structure of membrane proteins is most likely to be published in a very high cited journals such as Science, Nature, or Cell. By using the techniques developed in my lab, I am confident that we have the right expertise and facilities in UL to look at other membrane proteins. So the next step would be to study those membrane proteins involved in communicable and non-communicable diseases such as cancer or bacterial pathogenesis.